So now we're going to be talking about harvesting and utilizing energy in cells. First of all, by how we capture this energy in photosynthesis, and then how we transfer the chemical energy into ATP during cellular respiration. First of all, photosynthesis is the process that captures and transforms light energy into chemical energy. And sunlight is the ultimate source of this energy. Photosynthesis is carried out by photoautotrophs using water coming up from the roots, carbon dioxide from the air, and of course sunlight. And one of the byproducts, important byproducts, is oxygen. So the energy that comes from the sun is called electromagnetic energy, and it is in waves, photons. Here you can see these waves of light. Now the shorter the wavelength, the more energy it has, and the longer the wavelength, so the wider the peaks of the waves are, then the greater the energy it contains. And so here's a range of electromagnetic energy, the gamma rays, which are the shortest and have the shortest wavelength and the most energetic, and then we have the longest with the lowest. In the center here, we have the visible light spectrum, very small sliver, and that's the part of the light that we see. And most of the light that reaches Earth's surface is in this range, and it is the main form of energy that drives photosynthesis. So pigments are special receptive molecules that capture and absorb energy and light. Chlorophyll, carotenoids, they're a kind of chemical that does that, a pigment, but there's other pigments as well. Absorb energy that moves electrons to high energy states called excited states. And that energy that's captured by these electrons is transferred in a very orchestrated series of reactions that ultimately lead to the production of ATP. This series of reactions is photosynthesis. And this is just to show you an atom, and they're moving that electron from its ground state. Light energy comes in and puts it up into this more excited or high energy level. Now, different pigments absorb different wavelengths of light. So here you can see there's our chlorophyll B right here, and you have chlorophyll A. And you can see they're absorbing in actually opposite sections of the visible light spectrum, down here in the blue area, and then also up here in the red, yellow, orange area, not in the green area, which is kind of interesting, right? So the color of the pigment reflects the wavelengths they reflect and do not absorb. And so chlorophyll A and B are absorbing in this blue range, absorbing in this red range. They're reflecting in this green range, which is why we see plants as green. So chlorophylls are the major pigment molecules used in photosynthesis. Carotenoids are accessory ones. They also transfer energy to the chlorophylls, and we see these different pigments all around us. So here's chlorophyll. We have lots of carotenoids here in the plants that are changing color. Their leaves are changing color in the fall. And there's lots of different pigments in fruits, and they include things more than just chlorophyll and carotenoids. There's also things like anthocyanins as well. So keep in mind that pigments are special receptive molecules that capture and absorb light, this electromagnetic energy. And so photosynthesis, this is the basic reaction where we have carbon dioxide and water with this light energy giving rise to glucose and oxygen. And there are two steps to photosynthesis. There's what's called the light-dependent reactions, and they're called the other ones are called the light-independent reactions. They used to be called the dark reactions, but that was bad because light-independent reactions actually do happen in the light. And this process is called the Calvin cycle. So what this figure is showing is a chloroplast, and it's showing that both of the parts of photosynthesis are happening within the chloroplast. The light reactions are interacting with the thylakoid membranes here. The Calvin cycle is happening in the stroma. So basically in the light reactions, we have sun energy is being captured by the pigments and converted into the chemical energy of ATP, which is energy, as well as NADPH, which has some reducing power that helps to power that Calvin cycle, which is ultimately going to produce sugars. So you can see ATP and NADPH are the main products of the light reactions that help to power the Calvin cycle in the production of sugars. In the process, of course, ATP becomes ADP and NADPH loses its electrons and it becomes NADP. In the Calvin cycle, or the light independent reaction, ATP is used to convert carbon dioxide into the carbohydrates. And of course, they're going to become other organic molecules in the plant. So here is the light reaction, so the light energy, and here is our thylakoid membrane here. So remember, they're all stacked up in what are called grana. And it's just another image to show you the ATP and NADPH are the important products of this light reaction to go into the Calvin cycle. Now, I don't want you to get all worried about this illustration. If I ask you anything about it, I will give you this illustration on any exam. But what I want you to notice is we have this membrane, this thylakoid membrane, that's a bilayer, a phospholipid bilayer. It's got lots of proteins associated with it that are important for photosynthesis. We have light that's being harvested to excite electrons to ultimately produce NADPH. But we also have other things happening where we have the splitting of water. We build up a concentration of protons to the interior. And so we have a passive transport of these protons through the ATP synthase complex, which helps to generate the ATP. So this was a good figure. Go back to the video because the fellow there explains it really nicely. So here's an overall diagram showing you what goes in, what comes out of the light reactions, as well as the light independent reactions. And remember that the ultimate product is glucose, which is an organic molecule, which is basically the building blocks of other organic molecules, including proteins, very important, lipids, and really important for the production and growth of plants and animals. Now, the oxygen that's released is required for cellular respiration and the transfer of energy from organic molecules to ATP, which is what we're going to talk about when we talk about cellular respiration later on. But first of all, let's take a look at that Calvin cycle. And I don't, again, want you to know all the stages and the chemicals that are involved. Let's just look at the basics. So basically, the Calvin cycle occurs in the stroma of the mitochondria, of the chloroplasts. And the most important things are that carbon is fixed, and it's fixed using this enzyme, rubisco. And rubisco is the most abundant enzyme on the planet. And the first product is a C3 molecule. So that's why this is called C3 photosynthesis. Okay, the first stable compound is 3-carbon. So we have the ATP and NADPH coming in from the light reactions. And there's 3 stages. We have the fixation of that carbon, we have the reduction, but of course we also have regeneration because this is a cycle. So we have to have the regeneration of the components of that cycle. And we also have, of course, coming off that cycle, our byproduct, which is going to be important in the synthesis of organic molecules. Woo-woo. Okay. So there's our ATP, our NADPH, synthesis of sugar. ATP is required for the recycling of some of the compounds in this Calvin cycle. And here's a nice illustration. I'm going to give it to you as an extra handout. It's just something to take a look at in a bit of different perspective to help you get a handle on these 2 processes. So let's take a look at cellular respiration. It's a pathway that harnesses and uses the chemical energy stored in organic molecules to generate that ATP. And plants and animals both use organic molecules, including glucose, as well as oxygen. And you can see that it's kind of the reverse of photosynthesis. We have glucose and oxygen giving rise to carbon dioxide, water, with the production of ATP. And cellular respiration is a 3-step process. We have glycolysis, citric acid cycle, and oxidative phosphorylation. So glycolysis, well, that's easy. Glycolysis means splitting of sugar. So we have our sugar molecule here split into pyruvate. And it occurs in the cytoplasm of all cells. So it's a series of enzyme catalyzed reactions that break down glucose, which is a 6-carbon sugar, into 2 molecules of pyruvate. And it generates NADH and ATP. So here's a mitochondrion. Remember, glycolysis is occurring out in the cytoplasm, but the other 2 processes are happening inside of the mitochondrion. Eukaryotes use mitochondria to complete cellular respiration. The citric acid cycle occurs in the mitochondrial matrix, which is inside both of the membranes, so right in here. And then we have the oxidative phosphorylation occurs on the inner mitochondrial membrane, kind of similar to the electron transport chain that we saw in photosynthesis. So there we go, the inner mitochondrial membrane right there. And there's our cristae that we talked about before. So here we have the whole process. We have glycolysis, which converts glucose, a 6-carbon molecule, into pyruvate, which is a 3-carbon molecule. Only 1 chemical bond is broken, so we get some energy from that. But pyruvate still has a lot of energy because it's still got a whole bunch of carbon-carbon bonds. We have pyruvate oxidation to give us acetyl-CoA, which is what gets fed into the citric acid cycle. So, in the citric acid cycle, the rest of the bonds are broken, and we have lots more energy right here. Okay, but we also have a couple other products right there. Okay, and so the energy that's released is also harvested and held by NADH and FADH2. So those are 2 products there. And when we take a look at oxidative phosphorylation, we see those are our important donors for electrons. So these NADH and FADH are really important because they actually store the electrons that are going to be used in the process of chemiosmosis, which should be familiar to you, that creates an energy gradient that powers the enzyme ATP synthase. So these terms are familiar to you because we saw them when we took a look at photosynthesis, at the light reactions. And this ATP synthase, of course, is going to generate ATP by a different process, a little bit, it's called oxidative phosphorylation. And it's called oxidative because it actually requires oxygen to receive the electrons. Phosphorylation, of course, because it leads to the addition of a phosphate group to ADP to produce ATP. And this is the level of detail I want you to know, but remember that this is happening on the inner mitochondrial membrane. If you take a look at figure 5.20, you'll see that some of the processes are very similar to the light reactions that we saw in photosynthesis. I don't really want you to know the details of this diagram, that's why I haven't blown it up any bigger. This is kind of a nice flow diagram to show you the different components, what goes in, what comes out. Theoretically, and under optimal conditions, 38 ATP are generated by oxidation of 1 glucose molecule. But things are not always optimal, so in reality the value will depend on the cellular conditions. So, you know, it's not just glucose that is a source of energy. There's 3 major categories. Carbohydrates, of which glucose is one of them, it's a monosaccharide, but also other monosaccharides such as fructose and whatnot can be used as sources of energy. Polysaccharides, including things like sucrose, starch. We have proteins that are made up of polypeptides, of course made up of amino acids. And we also have lipids that are composed of fatty acids and glycerol. They can be interconverted from one to another, and they can be broken down into various components that can come into the process at different places. So for example, amino acids from proteins can be converted into pyruvate or acetyl-CoA, or actually be intermediates in a citric acid cycle. So they're coming in at different places. Same thing with glycerol from the fatty acids, from fatty acids. They can be converted into various components, as well as glucose to go into glycolysis. Fatty acids can be fragmented and can go into the pathway here as acetyl-CoA. And all of the arrows here can also be reversed as well. So some things can be precursors to other things. So, for example, pyruvate is used in the synthesis of amino acids. And if we think about it, cellular respiration is linked to photosynthesis. We have the production of glucose in photosynthesis, or those organic molecules that get broken down into cellular respiration, giving off carbon dioxide and water, which can be used in photosynthesis. So there's a link between those two. Cells undergo both photosynthesis and cellular respiration. They happen to do more photosynthesis because, of course, the glucose molecules are being used to do other things. They're not just being used in respiration. They're used as building blocks for cell walls, for enzymes, all sorts of different things, right? And so we have a higher rate of photosynthesis than we do at respiration. And different parts of the plant, of course, are going to be doing different amounts of each of those processes. Animals, of course, can only undergo cellular respiration. So this is a lovely little diagram just to show you the interconnection between photosynthesis and cellular respiration. Good study guide, I'd say. Okay, well, that's all she wrote, folks. Thank you for your understanding and letting me do this, and we'll talk to you later. Bye for now.